Our first presenter is from MaxCyte. Joining us is Dr. Linda Meggiolaro, Field Application Specialist. Welcome, Linda.
Hello everybody. Thank you very much, Perry, for your introduction. Thanks for all the organizers for making this event possible, and happy birthday as well, I would say. I would like to start today's presentation with a pop-up question for you all. When did you start to think about on and off target risk assessment strategies? Feel free to answer. The reason why I'm asking so, it's because today's presentation will focus on SeQures and how we can support cell therapy development from concept to clinic through regulatory success. Our disclaimer. Before starting talking about regulators and safety, I would like to briefly introduce who we are. MaxCyte has been in the cell and gene therapy field for the past 26 years. And since the beginning, we focus on developing and enabling safe and efficient cell electroporation. Our expert electroporation technology has been known for being application agnostic, and it enables to scale the production of your therapy from concept to clinic in a single platform. You can see it here. You may know that our expert technology is supported by an FDA master file and regional equivalents. Indeed, as you can see in the image, experts support several clinical trials and has been commercially validated in Casgevy since 2023. But what you may not know is that MaxCyte is also able to support and streamline the safety validation of your therapy with SeQure. SeQure is our new gene editing risk assessment service that has been founded in 2020 from Keith Young, and which is a leading name in the field of gene editing. We all know that when it comes to gene editing, it is very important to understand the on and off target events editing to prevent and avoid any potential and deleterious outcomes. The main goal of your therapy is to have an high potency. So it's to have on-target events in your wanted mutation site. But it's really crucial to understand the potential off-target mutation and their consequences when the therapy is administered to the patients. So for these reasons, to preserve patient safety, a therapy is going through a multi-step and strictly regulated development process. Everything usually starts from a scientific idea that must be, first of all, tested through discovery phases and tests. It goes through a preclinical and then clinical tests before to be commercially manufactured for patient's treatment. The entity that regulates and evaluate the safety of your therapy are a regulatory agency. Every country and region put in place a specific agency that, in order to fulfill their function, generates guidelines and standards. So if you're a developer and you are aiming to develop a therapy, we really recommend to connect with your target regulators as soon as possible in order to start familiarizing yourself with the guidelines and the processes that are required. In the context of off-target analysis, the FDA is the only regulator that requires a single and comprehensive guidelines. You can see here on the left. Outside the US, instead, the guidelines are more fragmented. You can see two examples of guidelines that are recommended from EMA in Europe. But overall, the expectations is that it's developer responsibilities to put in place a solid and comprehensive off-target risk assessments that best characterize your therapy. During these safety assessments, you want to be able to identify the editing efficiency of your therapy, but as well, you want to identify the off-target events and chromosomal rearrangements that are generated by your editing tool. To do so, you want to leverage in vitro, biochemical, and in cellular assays, so orthogonal assays, and some of them should consider different genomic populations. It's also the developer's responsibility to be able to interpret the assays, the findings. You want to be able to understand whether the benefits of your therapy and whether your therapy has a potential risks for the patients. All the findings should be then rearranged and presented, delivered through a comprehensive report that should evolve through the different steps of the development of your therapy. So it's not a single exercise only. You should progress and repeat this safety evaluation also through the clinical phase and post-marketing authorization. How can you achieve that? Well, SeQure offer 8 different assays. All of them are meeting the regulators' requirements, and they can support you through screening, nomination, and confirmation of off-targets. SeQure's assay mostly wants to address 2 main questions. What may potentially happen in your cells when you leverage your therapy, and what actually does happen in the cells when you leverage your editing tool. If you're in the first phase, you can start through a screening analysis. For example, you're a developer that has a list of potential guide RNA sequences, and you don't know which one to choose to move forward with your therapy. So you can use Guide Profiler and Guide Select to screen and rank your guided RNAs according to their safety in order to mitigate risks and potential delays during your development phase. So with your favorite candidate, you can then start the nomination phase, leveraging OneSeq, GuideSeq, ChainSeq, and DoSeq. So those are all orthogonal assays that will address the same questions. They will enable to enumerate potential off-target sites caused by your editing tool. Those off-target sites will be then validated through a confirmation phase. So will be validated with AmpliconSeq and safer detection. And you will be able to confirm the bona fide of targets observed in your edited cells. In this table, you can see a summary of the properties for each assays and their main uses. There is a lot to be discussed, but I want to highlight that Guide Profiler, Guide Select, and OneSeq are the variant-aware assays for screening and nomination. OneSeq, together with DoSeq, GuideSeq, and ChainSeq, enable nomination of off-targets. They are all orthogonal assays, and we normally help developers to understand which assays best works for their safety validation according to the type of editors that you want to validate and the cells that you want to edit. For example, if you are developing a cell therapy where you want to edit HSCs with base editors, we may advise you to run OneSeq and ChainSeq because both assays are compatible with base editor. OneSeq is variant-aware, and ChainSeq it's unbiased and as well compatible with the sensitivity of HSC cells. To conclude, AmpliconSeq and safer detection are able to validate bona fide of targets, specifically indels and chromosomal rearrangements. Among the assays that SeQure offers, there is OneSeq. I just mentioned that it's really important and crucial if you want to do variant-aware nomination studies. How can we achieve that? Well, OneSeq is not just an in silico assay, it's a biochemical one because it consists in 2 main steps. There is first an in silico search across thousands of different genomes to be able to enumerate off-target sites, so sequences that are very similar to your on-target sequence. And this list of potential sites will be then validated in vitro through a biochemical cleavage using your editor. So at the end of these assays, you will be able to identify the impact that genomic variance has in terms of off-target events. Why is this important? Because when you are assessing the potency, the efficacy, and the safety of your therapy, you want to do so by taking into consideration the all intended to treat population and not just a few donor genomes or like a few genomes from patients. I also want to mention that in case you run a guide profiler and guide select for your screening phase, you will be able to run a computational search that will have the same variance-aware capabilities than OneSeq. In these slides, you can see as well some articles that are showing you how OneSeq, GuideSeq, and ChangeSeq all were able to support IND filing applications. Just for clarification, OneSeq and GuideSeq are the original assays invented by Keith Young, while ChangeSeq instead has been invented in Shankar Lab, which is Keith's former postdoc. As well, for every assay that we run, we will deliver a comprehensive regulatory-ready report that is going to detail all the potential biological impact that the findings means for your therapy. In the center of this slide, you can see an example of a plot where off-target sites, so all these green dots, have been nominated using OneSeq. And these sites have been categorized in tiers 1, 2A, 2B, and 2-3, according to two main parameters, the frequency of these assays and the genomic annotations. Normally, we recommend to move forward during your validation step, considering off-target assays located in tier 1, 2A, and 2B, but you're also flexible to include extra off-target site in case you want to have a more comprehensive and extended validations. So with this presentation, I hope that I gave you a flavor of what SeQure's assays can offer. But of course, there is so much more to be discussed, so feel free to reach out directly to myself or your regional MaxCyte FAS or directly with our website. So thank you very much for your attention, and I'm happy to take any questions.
Thank you, Linda. Great presentation. You spoke about this, but so if I want to perform off-target nomination using two of your assays, for example, a OneSeq and a ChangeSeq, so the results that you provide, are they just simply results for each, or is there a comparison that you do?
By default, what we like to do is to generate a single regulatory-ready report where for every step, so I just show you the tiers of off-target results, but then also we will look at the on-target sites, how frequent it's going to be, if it's going to be close to the on-target site. So we usually, for every point of the assays that we check, we will generate a single report where we compare the findings for OneSeq and ChangeSeq. So it's going to be, so there is not any extra additional work that the developers should be doing in order to submit these to support the IND filing, but as well we can be flexible and we can generate a report for each assay in case the developer actually is prefers so.
Okay, very good. So you introduced and spoke about three assays that consider population variance at on and off-target sites. Are all in silico assays?
So it's a good question. So Guide Profiler, it's the only fully in silico assays that we have, while Guide Select and OneSeq are biochemical assays. I try to explain it a bit for OneSeq. So OneSeq and Guide Select are biochemical because as Guide Profiler, so the first assay, they will run an in silico search considering thousands of genome as a reference and not just the HG38 as a reference genome. Guide Select and OneSeq will then be followed by an in vitro cleavage because we want to validate the enumerated loci that are similar to the on-target sites, and we want to validate it in vitro in order to be a bit more specific and not go and enumerate thousands or hundreds or thousands of sites that then you don't know what you should prioritize when you move forward, when you assess the safety of your therapy.
Well, thank you. Great explanation and clarity on that. So thank you, Linda. Great presentation. And thank you for being with us today, Dr. Linda Meggiolaro from MaxCyte.
Thank you.